Hello, this is Rob Durham,
And this is John Chappell.
and you're listening to another episode of the Gyrolab Spin. Well, on today's episode, John, we have a new edition where we're trying out video. We got, by popular demand, there was an insistence that we have our video on.
Yeah, people really want to see what we look like for some reason, and
so the headshots from last year, I haven't aged that much, hopefully, but I have been told I have a face for radio, so here you are.
yeah, but you are the company Barry White, Rob.
Excellent. Okay, well, thanks for joining us on this next exciting episode. On today's episode, we're going to have a second discussion about affinity on the Gyrolab system, and what we wanted to focus on in today's discussion is about a practical guide to really setting things up on the Gyrolab.
Yeah, I mean, this episode really is aimed at bioanalytical scientists who aren't basically that familiar with affinity or haven't really had a background doing like a BIA core or SPR, and to really show them how they can utilize the system and actually get really good affinity data. And it's a real advantage of the platform that you can generate this data, and it can be a very useful application, particularly when you want to characterize reagents or you want to look at your binding of your analyte, etc. So it's a very useful aspect to be able to use the system for.
Absolutely, and I think one of the things we wanted to highlight today is that if you're able to do a PK assay on the Gyrolab system, you can certainly set up and analyze data using the Affinity module that comes on the Gyrolab system in a pretty straightforward way. You don't have to have a lot of extra expertise in doing affinity measurements that you might have with other platforms.
And I think that's the key here is we will go through how to set up an affinity experiment, and hopefully then you can apply the same principles to your method.
So here, this is a slide from the earlier presentation that we did, which just gives you a basic overview of what affinity is. And again, in a very simplistic way, it defines the overall binding strength of an interaction between two interactants. So in this case, we have A and B, and then this is described by the on rate versus the off rate, and a KD is really the ratio of the off rate to the on rate. And so that's kind of what we're dealing with here. And I think the important thing to remember from an analytical perspective is that KD is typically described in molar units rather than in mass units, and so we're converting things into moles.
I mean, that's critical when you set them an affinity experiment, that you need to convert from mass units, because as bioanalytical scientists, we normally work in nanograms per mil, etc. So you need to convert everything into molar units. And I think the other thing really important is that if you have really high affinity, it's going to take you a longer incubation time to reach equilibrium, and that's quite important actually when we start designing the experiments.
Yeah, I think the thing about the Gyrolab system is that, again, remember that the equilibrium happens offline. It's pre-incubation on a plate, and that gives you the flexibility to let that incubation go as long as is required to get to equilibrium, because that's when you're going to make your most accurate determination of KD.
So affinity is used throughout drug development. It's used all the way from discovery, research. It's very much used to define the interaction of your analyte to its target, and as a way of how, particularly when you're developing candidates, how to rank them, for example. So it's a particularly important as to actually look at how well your drug binds to its target. So it's very important. It's important throughout the molecular journey of the molecule as it goes through drug development.
And that's true from a molecule or a drug development standpoint, but I think it's also true for reagents. So if you're looking at screening reagents for other immunoassay applications, then again, finding those molecules with a relatively higher affinity is generally what's preferred.
Yeah, and also when you're making the drug, it becomes a key quality attribute when you're making different batches to make sure that your batches continue to bind with a similar affinity.
Right.
So now we're really going to move forward and go through our practical example of affinity determination to give you key aspects of how you design the experiment and how you set them up on the Gyrolab system. As a short recap for in-solution affinity determination, which is done on the Gyrolab, the kind of key thing is that very high affinities can be determined. And as we will show, neither interactants are modified and the interaction is performed in solution. So this mimics the biological system and biological activity. For example, when you're looking at things like surface plasma and resonance, for example, you're immobilizing interactants on the solid surface. So that might not reflect the biological activity, although it is the gold standard for affinity.
It often is. And the other limitation of that application or that particular platform is that those off rates are exceedingly slow the higher the affinity is, and so you have to go to very, very long experiment times and see very tiny differences, and that can lead to error in your assessment of the off rate.
Yeah. And when it comes to in-solution determination, Gyrolab is a flow-through microfluidic system where your actual reaction time is in your seconds. So there's very little chance for the interaction to dissociate as part of the assay step. And that's an important part of the affinity determination on the system.
Yeah, and I think a number of people would have done this kind of work on a typical immunoassay plate, and so that's fine as long as the affinities are relatively low. But if you have, again, high affinity interactions, then you're going to end up having longer incubation times, and that will lead to potential equilibrium shift within the plate. So here in this example, you can see how you'd use a PK curve on the left, where you're using your standard calibrator series and seeing an increase in response relative to an increase in the free concentration. And what the circle represents is our selecting one of those points on the curve that we can then use as the starting point for doing our affinity curve.
So that determines your fixed concentration, and then you vary the concentration of, in this case, the drug, and then you inhibit the signal. And this is used then to define an affinity curve, which then will look at your binding constant, effectively your KD, from the middle of the curve.
And so we're using the standard immunoassay format, but we're using a slightly different experimental design, and we'll get into the nuts and bolts of how that all works in subsequent slides.
So after we've done our concentration curve and you've kind of worked out what sort of fixed concentration you're going to do, you then need to prepare your affinity sample. So as mentioned, we mix a fixed concentration of one interactant that we've chosen from the previous experiment with a variable concentration, the binding partner. So effectively you look at different concentrations against a fixed concentration, and then you form an affinity series in the microplate wells. Because you're actually interacting offline, there is no labeling of the interactants. What you're effectively doing is trying to measure the free amount of fixed analyte. We will go through it using our kind of our test assay. We're going to go through the IgE and specifically show you how this is practically done, giving you actual examples showing how the curve and how we've decided on the concentrations to use.
And I think this is a place where it's helpful to know about the range of your KD ahead of time, but if somebody doesn't, they simply have to do an experiment with a screening at a specific time point, and you'll be able to see sort of a rank order of your molecule KDs, and then can adjust as you want to refine what your KD measurement is. But if you just pick, say, for an hour as an incubation time, as often as done, then you'll be able to screen a range of different compounds and then come up with an estimate of the KD, and then can refine that based off of the data that you get from the earlier experiment.
Yeah, and I mean another good example is you can do your affinity samples and you run them after say 2 hours, and then you can also then leave the samples to incubate overnight.
Yeah.
And then you can see if there's any shift, and if it hasn't reached equilibrium, then you know you have to incubate for longer.
Yeah, you'll see a shift over up to, we've done experiments of very high affinity interactants that go up to 3 days. Again, that's something we'll have to optimize as you move forward, but certainly could be done in a screening mode at a shorter incubation time.
So now moving over to our specific example, where we're looking at Xolair, which is a monoclonal antibody that binds to human IgE. It's an important treatment as an anti-inflammatory and kind of anti-allergy treatment. The drug actually inhibits the binding of IgE to its receptor, so on mast cells and other cells. So it desensitizes the ability for allergens to cause an allergic reaction. So it's actually quite an important treatment. Kind of an important part of the affinity of Xolair to IgE is actually it's relatively low affinity. So this has very much become our test system. Because it has low affinity, it means that when we're doing this assay for training purposes, we only need a couple of hours of incubation to reach equilibrium. So it makes it a very good assay for training purposes.
Yep, and I wonder, it sort of speaks to the question of it isn't always the highest affinity drug that's the most therapeutically useful. So sometimes a slightly lower affinity has a better biological efficacy profile, and this is certainly the case for Xolair.
Exactly, and I think what the other thing that shows us one thing about the interaction of Xolair and IgE, that if you dilute samples, for example, you may get a dissociation of the complex. So this is actually quite a good assay to analyze on the Gyrolab, particularly if you want to look at free IgE. We've developed assays in 100% matrix to measure free IgE, and effectively that's using a part of the kind of the affinity concept on the platform, where you're getting low dissociation as part of the assay.
Yeah, and the idea that you can do it in 100% neat matrix is a real advantage in this case, because any kind of dilution of the sample is going to sort of re-equilibrate your free.
Exactly.
So this is just an overview of how we set up the Gyrolab assay using the Gyros method. Here you can use either a 200 or 1,000 nanoliter CD on the instrument. And again, the selection of CD is dependent on the sensitivity of the assay that you want to devise. The more sensitive the method, though, that will allow us to measure a higher affinity interaction. So the higher the affinity interaction, we're going to have much lower free interactant available, and so you need a more sensitive assay for that. But again, it's a standard method. We have our biotinylated variable interactant, which in this case is Xolair. Our free fixed is bound to the captured variable interactant, and in this case that would be IgE, and then we'll detect by a fluorescently labeled anti-IgE detection molecule.
Just to comment on the CD, actually the IgE assay uses 1000 HC, but the assay has been run on every CD from 20 all the way to 4000, and it really depends on which sensitivity you need, and generally we run on 1000 HC. So as kind of already indicated, you need to decide how you're going to determine your concentration of fixed analyte as part of the assay. So as mentioned, we try and as a minimum go 10 times the signal to noise to the signal to blank. So, and if you then I would probably recommend that you run the experiment with various concentrations using 10-fold for blank, and then go at least 5 to 10 times higher, and do at least 2 fixed concentrations for affinity determination. If you go all the way up to say 50 to 100 times concentrations, you'll actually go more to determining the f-top determination, where you go away from a KD control curve.
Yeah. So in that stoichiometrically controlled situation, you're going to more accurately determine the active concentration that you're working with. So again, it's a hunting expedition a little bit to sort of sort out. But if you start at tenfold over background and then you incrementally titrate up towards 50, you'll probably hit a couple of points that will be reasonably good KD controlled curves that will give you some wiggle room in terms of hitting a reasonable KD.
Yeah, I mean the software will allow you to run affinity curves and actually pull the data and actually visualize all the data from all the fixed concentrations. Because I know from it, kind of some of the training sessions we've done, the more fixed concentrations you can choose, the more chance you've got of determining the KD first time.
Exactly. And again, there might be different ways that you want to set up your experiment. If you're really just screening, you might have one concentration for a lot of different molecules. But if you have one molecule really trying to dial in the affinity, then you can run that one molecule at several fixed.
So moving on to our practical example. So we're looking at the IgE, and we start off with the concentration curve. So we will determine what fixed concentration we will apply to the affinity experiment. So we take the normal IgE assay range is in international units per mil. So the important thing is that you convert your concentration units to molarity, so into picomolar in this case. And we've run an extended curve. And then we then run the IgE concentrations using the biotinylated zone layer for capture, and then with detection with anti-human IgE. So this will be used to determine the fixed concentration, and then we'll go through how we've done that and the data that has been generated.
And so this is really, in essence, a PK curve for IgE.
Yeah, the difference is we've just converted the units to molarity rather than the normal mass units that you would use if you were doing a PK assay.
So here in this example, we've actually taken our IgE titration curve, and you can see then at about 20 picomolar, we're at tenfold over background. And so that gives us a starting point for where we can begin to do the fixed concentration for our IgE in our interaction curves.
Yeah, so I'd probably suggest that you do at least a fixed concentration at 20, but then you also look at least 5 to 10-fold higher. So you can compare the data to maybe 50 to 100 picomolar and run at least two fixed concentrations.
So once you've selected your fixed concentration, the next thing you have to do is figure out what the variable concentrations you're going to pick. So again, you're going to pick a range of variable concentrations that should cover the KD as well as achieve an asymptote on either side of the expected KD. So in this green curve illustrated here, your KD is going to be somewhere in the midpoint, and you want to be sure your concentration range gets us up to both the upper asymptote as well as the lower asymptote. And so the sort of rough rule of thumb is that the highest variable starting at about 50 times the expected KD. And again, if you don't know what the KD is, you're kind of left sort of, taking your best guess at this. But the other thing to consider is how many points do you want in your curve. The Gyrolab system is flexible and it will let you use any number of points. I would say typically 8 would be on the low side, and 12 is something that I've used typically that works out well on the CD. You do 1.5 segments for 12 points or 16 points. So again, if you're using a higher number of points, you can do smaller dilution steps, or if you're using fewer points, then you'll have to do larger dilution steps in order to cover that broader range of the curve.
You normally would prepare the top standard and then you apply a consistent dilution, say a 1 in 3, 1 in 4, to ensure you get a wide range. And depending on what you achieve, obviously it is important that you define both the upper and the bottom parts of the curve, because it is a curve fitting model, so it requires a good curve fit to calculate an accurate KD.
That's good, that's right. And then the other sort of practical thing that's helpful is if you can keep those dilution steps the same across the curve, that makes it much easier to set up your curve. Whereas you really have to put your thinking cap on if you're pipetting and you're doing 10-fold dilutions and then 5 and 2 and then back to 5 and 2 again and 10. It can be more confusing to set it up that way. So,
and also increases the potential error when you're doing the preparation as well. So this really shows your experimental design, and it really conceptualizes the concept of affinity. So as I've mentioned, we've determined the IgE fixed concentration, and then we vary the concentration of Xolair, the binding variable interactant. And as you can see here, as you increase the Xolair concentration, you'll decrease the IgE free measurement. So what we're doing here effectively is measuring the free amount after it's reached equilibrium, and this allows us to calculate the kind of the KD. And this really shows you, how the affinity part fits into the assay measurement.
Exactly. So here, if we're set up for about 1,000 nanomolar as a top variable Xolair concentration, then to make the standard curve, we would start at 2,000 nanomolar because we want to have a 2x of our standard curve so we can combine the fixed and the variable interactants together.
Yeah, doing a one-to-one dilution is the much easier way of actually doing this experiment.
Exactly, and so then you'll make your standard curve points down 2-fold dilutions for 16 points. So we've got a lot of points on this case, so we can go with a lower dilution factor and still cover the range of variable interactant that we need. And then if we've decided on a fixed IgE at a final of 200 picomolar in this example, then we'd make a stock at 400 picomolar, so that 2x concentration, so that when we add our fixed concentration to each of the 16 wells that have a variable concentration of Xolair in them, then we're going to end up with a final concentration in the well that is half what we started with in our stock.
Yeah, so the 1,000 nanomolar Xolair is approximately 50 times the expected KD. So that's kind of the logic behind that starting concentration. And as mentioned, we know that the affinity is reasonably low, so we incubate for 2 hours at room temperature and to actually perform this experiment.
So here you can see what it looks like in the Gyrolab analysis window when you do a single point affinity measurement. You can see, again, as expected, as the Xolair concentration increases, we're getting a decrease in the signal shown in the window on the left. Now, while it's a little difficult to see on the slides, there's tables on the right which show the calculated KD values as well as the FTOT values, and you can determine how accurate those are. And then the table below shows each of those data points, which illustrates the response value for each of the replicate values. In this case, we had 2 replicate points for each point in the curve.
Yeah, one kind of important point is that it's about overall modeling. So you want to make sure, as we've said earlier, you want to define the upper and the lower end of the curve. And you a lot of the time you will see a lot of scatter, but particularly at the really high responses, because you're talking about really little changes. So do not be concerned about this. It's the overall model that matters.
Yeah. One thing I'll often sort of remind myself is that this is usually a linear scale on the y-axis. Normally in our curves, in our PK curves and things, people are looking at very wide concentration ranges. So they'll often have that be a log scale. And so you're used to seeing very tight overlapping dots. And so that's why I'll often use the table at the lower right. First thing I'll do is check to make sure there isn't anything that's wildly out in terms of CV between the replicates. And if I see that, then I'll, again, as I normally would, check the viewer profiles to make sure that the images look good. And if all is good and I don't see any wildly out CVs, then I'll be able to proceed with the analysis.
So here's another example. So we've actually run the IgE at 2 fixed concentrations at 20 picomolar and 100 picomolar. as mentioned, we incubate for 2 hours, and then we actually do a comparative KD calculation, and you can see that the software can normalize the data, and this allows you to view all the fixed concentrations and actually see the difference. And in this case, you can see with the 2 fixed concentrations, we get a really good calculation of the KD, and it's very consistent.
Yeah, I think that's the other point, and we'll bring it out later. But in many folks who are inexperienced, as I was when I first started doing affinity on the Gyrolab system, you're concerned that you're seeing pretty different numbers sometimes between runs or between different methods. And it's important to remember that with affinity, as long as you're within sort of the same ballpark, not in the same exact space, it's a different measurement.
Yeah, and as we mentioned in the previous podcast, as a bioanalytical scientist, we like to see within 20% or whatever reproducibility, but that isn't necessarily appropriate for affinity. It's about being low picomolar, for example, or very consistent nanomolar measurement. So if you've got 10 or 50, it's pretty much the same, for example.
So again, here we've got a table which talks about variability in interpreting KD. So again, you get here's from a paper that shows 150 different participants using the same protein samples, 50 kilodalton Fab versus a 60 kilodalton GST antigen, and you can see that you get quite different numbers across the different labs and the different platforms. And so again, this is something that's to be expected.
Yeah, and as we said in the previous slide, it's really about being in the same ballpark. So absolute number doesn't matter. It's a relative, and it's actually it is method dependent. It is per day dependent. You will get variability in your measurement.
Yeah, I think again to point out that maybe the obvious, but I mean, since it's a relative measure, if you're screening, a series of compounds and you care about the differences between the two, as long as they're screened on the same day under the same conditions, you should get pretty good rank order affinity measurements using the Gyrolab system.
Thank you, everyone. So we've kind of given you an overview of in-solution affinity determination and given you an example and shown you how the method was set up and how the decisions were made about the concentrations of both fixed and variable concentrations. I mean, it is important that you actually set the experiments up correctly, apply good scientific principles, and the good thing is that the system is very flexible. You can do your incubations offline, so you're not necessarily occupying the system while you're doing your KD calculations. So, and as I said, you're not modifying interactants, so you do get a good indication of the proper biological activity, and the microfluidics will minimize equilibrium shift, which can be very important during the measurement.
And I think the other thing that's really kind of useful is because you could set it up and run an experiment multiple times from the same plate, essentially. You because it uses very little sample, you could essentially rerun the same affinity series multiple days and determine whether you're getting that much of a shift or not, and whether you've achieved sort of a plateau in the KD values that you're getting.
Yeah, I mean, that's an important point, that the Gyrolab system uses very little sample volume, so it does give you flexibility to really design your affinity experiment over multiple days.
Excellent. Well, I hope this was helpful for folks out there.
Yeah, please send us any comments if there's any help that you need, and hopefully we've given you confidence to use the system to calculate affinity. Just a quick acknowledgement for people within Gyros Protein Technology. We have our resident affinity experts both in the US and Europe. So Ellen, who is based in the US, is very much one of our leaders in the affinity calculation. The same with Linda, who's based here in Europe. She very much has worked with many customers in affinity calculation. And Johan Engstrom is our internal affinity expert, and we will getting onto a future podcast to maybe go through some more advanced topics in affinity, because there are advanced topics.
Yeah, I think people that are doing on cell based affinity, and then if you have multiple
binding sites,
that's always a fun topic to sort of get your mind around when you're trying to do affinity.
So there will be an affinity part 3. So we look forward to that in the future.
Yeah. Well, that brings us to the end of another episode, John.
Yeah, and hopefully the video has not put you off attending any future podcasts.
That's right. Give us some feedback. Let us know whether you really like the video or whether you want us to go back to no video.
Yeah, but I mean, it's been fun, and really what we need from you is suggestions and feedback on our podcast. Please contact us on the email address as shown on the slide. And send us any suggestions that you may have.
Yeah, we're really interested in your ideas for topics for future podcasts, other things that you'd like to see, questions that you have for us. We're happy to interact with the audience here and make this a forum that's really useful for everybody. So with that, I will call this a wrap for today, and I'll say goodbye from me.
And goodbye from him.
And until next time.